Hey guys, we're going to go over the Chapter 7 concept map. We're going to start off with talking about the cell theory. Make sure you guys remember that the cell theory applies to all cells. So that goes for bacteria, plants, animals, anything that's prokaryotic or eukaryotic or constructed of cells. Make sure you guys are also aware of the different scientists that played a role, and again be able to pick out which parts of the cell theory belong in a multiple choice question. We may have 4 choices there. We might say or ask you which one does not belong or which of the above does belong. So just make sure you guys are aware of the cell theory and its key parts. Also again be familiar with the different scientists and the roles that they played in the experiments. There are cell types that we covered. The 2 main cell types we covered were prokaryotic and eukaryotic cells. Make sure you guys are aware of the difference between the two, and then again, what are some things that they have in common. Examples of eukaryotic cells are animals and plants. Examples of prokaryotic cells would be bacteria or blue-green algae. Bacteria is the more common one that we talk about. There are 2 things that really distinguish a prokaryotic cell from a eukaryotic cell. Eukaryotic cells have a true nucleus, which means that their DNA is protected by a membrane from the rest of the internal parts of the cell. Prokaryotic or bacteria do not have a true nucleus. Their DNA is in the cytoplasm with no membrane protecting them from the rest of the internal cellular environment. Both cells contain DNA, but again the difference is eukaryotes have their DNA protected by a membrane. Prokaryotes do not have their membrane protected, or their DNA protected by a membrane. Another distinguishing factor that separates prokaryotic from eukaryotic is bacteria and prokaryotes do not have membrane-bound organelles, where eukaryotic cells have many membrane-bound organelles, and each of those organelles has a certain function that plays a key role in the cell's activities. Also guys, make sure you are aware of distinguishing the different types of microscopes. The 2 main scopes are light and electron microscope. A light microscope allows you to look at live or dead organisms. It's the only scope that allows you to do that, so it's good for quick, raw observation of cells or organisms. The light microscope has limited in terms of its power, only about 1,000 times magnification, which is limiting when you're looking at small structures such as organelles or certain types of cells. The transmission electron microscope, you must look at something that is already dead. The specimen has to be dead. The advantage of this guy is it can be magnified hundreds of thousands of times its normal size, which allows you to really see size and structure of certain cells and certain organisms. Another type of electron microscope is called a scanning electron microscope, and that produces a 3D image of an organism or a cell. So that's important when you're looking at the surface of either cells or of certain organisms. So make sure you guys know the advantage and disadvantages of each scope. The advantages of a light microscope is it can be looked at as a living or a dead specimen. The disadvantage is it's limited in its power. The advantage of an electron microscope is it has a lot of power in terms of magnification, but the specimen must be dead. And the scanning electron microscope advantage is it allows you to see the surface of organisms in 3D. The disadvantage is just that. It's limited to just the surface and a 3D image. Let's now cover the cell structure and certain organelles. Remember, organelles are tiny structures that have a specific function inside of a cell. Remember that prokaryotes do not have membrane-bound organelles. This is unique to eukaryotic cells. However, the structures you see circled here in blue are found in all cells, both prokaryotic and eukaryotic. The cell membrane, which is found in all cells, Its job is to protect, and its job is also to regulate the transport of material into and out of the cell. This is one of the ways the cell can maintain homeostasis. Cytoplasm, which is found in both eukaryotic and prokaryotic cells, is where the chemical reactions take place. It's also where organelles are being held. Some people refer to that as the factory floor. Ribosomes are found in both prokaryotic and eukaryotic, and their job is to make proteins. Nucleic acid is responsible for the genetic instruction to make those proteins, and most cells have their DNA via the nucleic acid found. So these four structures are common to all cells. The ones below are more found in eukaryotic cells. The nucleus is a structure that contains a nucleolus, which is involved in RNA. That's a dark, dense structure you see, usually stained heavy under a microscope. And then the nuclear membrane, or nuclear envelope, which protects the DNA and surrounds your DNA. Again, this is what differentiates a prokaryotic cell from a eukaryotic cell, one of the main differentiating factors. Other structures or organelles, the endoplasmic reticulum is a series of tubes that allow certain particles to be transported throughout the cell. Particles that go through the rough ER, that is where proteins are modified. The rough ER is called rough because its surface is studded with ribosomes. The smooth ER does not have ribosomes studding its surface, hence the word smooth. And these guys modify lipids, drugs, alcohol, etc. You'll find a plethora of these in your liver because that's the job of the liver, to detoxify and partially filter out fatty material. The Golgi body, its job is to package material and then ship it out or export it by means of a vesicle. Usually one Golgi body per cell, and again they're found in both plants and animals, as is the ER and is the nucleus. Vacuoles are also found in both plants and animals, but plants have a large vacuole, animals have a small vacuole. The job of the vacuole primarily is to store water. There are some other jobs, but they're minor in its function for our purposes. Mitochondria is found again in both plants and animals, and is the powerhouse of the animal cell primarily. It allows the cells to go through a process called respiration, which ends up producing ATP for the cell. That's something we'll get into more at a later time. Chloroplasts are found in plants only. Their job is also creating energy for their organism, and we know that they go through a process called photosynthesis, will be the focus of our next chapter. Lysosomes, which are found only in animals, not plants, break down old organelles and bacteria. You'll find a lot of these guys in white blood cells, and they contain digestive enzymes that break down material, whether it's old organelles, whether it's bacteria, or whether it's food particles. These guys are also responsible for cell death, when the cell needs to terminate because of a mutation or faulty information. Cytoskeleton is found in both plants and animals. Cytoskeleton is involved in internal structure, just like your skeleton is. It's also involved in cell movement. You can find microtubules and microfilaments present here. Cilia are small bristle-like hairs, and down below the microtubules for the flagella, enabling the sperm to move. The last heavy topic for this chapter is cell movement. transporting material into and out of the cell. Again, we're talking about regulating, which means making sure not everything gets in or everything gets out. Hence the term selectively permeable or semi-permeable. The cell membrane is also called the plasma membrane. It's also called or referred to as a fluid mosaic model, which is another way that the cell membrane prevents everything from entering and leaving. Up here you look at your structure of phospholipids and the transport protein. Again, these guys act as barriers and preventative measures to help regulate what enters and what leaves based on the size and the charge of the particle. There are two major types of transport, passive and active transport. Passive transport is high to low concentration, so they call this moving with the gradient. Active transport is going low to high or against the gradient. In passive transport, since you're going down the hill, there is no energy needed. In active transport, since you're going up the hill, you're going to need energy, and that will come from the mitochondria producing a particle called ATP. There are different types of passive transport. Diffusion is where you're moving particles from high to low. Examples could be CO2 or oxygen. Facilitated diffusion is moving particles from high to low, but now with help from that transport protein. So something like starch, which is a relatively large macromolecule, may need help getting into the cell. But again, it's passive because there's no energy being used. And then osmosis is actually the diffusion of water, moving from high to low concentration. And depending upon the tonicity of the environment that the cell is in, such as isotonic is where there's the same amount of material in and out of the cell. In a hypertonic environment, there is more water outside the cell, so hypo will cause the cell to blow or swell because there is more water in, I keep on saying that, more water out of the cell, so it's going to rush in. Hypertonic is going to cause the cell to shrink because there is more water inside the cell, so it's going to want to leave and go from high to low concentration. There are different types of active transport. Endocytosis is bringing material into the cell by means of a vesicle. And exocytosis is moving material out of the cell by means of a vesicle. And again, try to always connect the different organelles and their structure. The Golgi body is going to make that vesicle to allow different particles to leave the cell. In the case up here, the lysosomes are going to help break down material that's being brought in, sort of like digestion and eating. Lastly, we have cell specialization. Our cells are specialized, having certain jobs that they have to do that no other cells can do. We are made up of a level of organizations, with cells being the smallest. A group of cells that work together make a tissue. Tissues that work together make up an organ. Organs that work together make up a system. And when a system of organs works together, that makes up an organism.